Yeah, good morning, everyone. My name is Cai Chun Zhou. I'm ISLAC President-elect. It's my great honor to moderate this press briefing that covers the second of Presidential Symposium, representing very best research from ISLAC World Lung 2024. Today's the Presidential Symposium press briefing covers 5 of top related abstracts submitted to the meeting. To house keeping notes, please hold all questions until the end of briefing, and please remember you can view the press briefing again on the ISLAC YouTube channel later today. Let's get started. First, I'd like to invite Dr. Yan from Guangdong Lung Cancer Institute, General Hospital, and she will present the osimertinib with or without sovinitimab as first-line in de novo MET aberrant EGFR mutant advanced non-small cell lung cancer, CTON 2008, the phase 2 trial. Dr. Yan, please.
Oh, thank you, Professor Zhou. Good morning, ladies and gentlemen. It's a great honor for me to be here and introduce a phase 2 study. It means proof of concept phase 2 study. Osimertinib with or without savolitinib as first-line treatment in de novo MET aberrant EGFR mutant advanced non-small cell lung cancer. It's also named CTON 2008 or flower study. Limited data show de novo MET amplification and MET overexpression occurred in 2 to 5% and 11 to 15% of treatment-naive EGFR mutant advanced non-small cell lung cancer, respectively. Preclinical studies demonstrated that coexistence of EGFR mutation and MET amplification or overexpression reduces the sensitivity to EGFR TKI, and is likely to be a key mechanism for mitigating primary resistance to first-line EGFR TKI monotherapy. Some clinical studies also shown that EGFR mutant advanced non-small cell lung cancer with MET amplification or overexpression treated with first-line EGFR TKI monotherapy had poor clinical outcomes. Dual targeted therapy with osimertinib plus savolitinib as highly selective MET TKI showed a potent anti-cancer activity in the lung cancer organoids drug testing assay, suggesting it may be a promising first-line treatment for such patients. Is the study design phase 2 prospective open-label 2-cohort randomized multi-center clinical trial in China, CTON 2008. Here I introduce the MET aberrance definition. MET overexpression is defined by IHC3+ in equal or greater than 75% of tumor cells. Also, MET amplification is defined by FISH or gene copy number of tissue NGS. All the eligible patients are randomized to receive osimertinib monotherapy cohort 1 or combination of osimertinib plus savolitinib cohort 2. Patients in cohort 1 will cross over to cohort 2 after resistance to first-line monotherapy. Primary endpoint is ORR. Second endpoint include DCR, DOR, PFS, OS, 12-month OS rate, safety, and tolerability. So baseline demographics and clinical characteristics were well balanced among the 2 cohorts. In MET aberrance, MET overexpression accounting for 87% and 67.2% respectively in the cohorts. The data cutoff date was May 28th, and median follow-up was 8.2 months. ORR was 60.9% and 90.5% respectively in the cohorts. DCR was 87% and 95.2% respectively in the cohorts. From the spider plot, you can see patients treated with osimertinib plus savolitinib showing deeper and more durable response over study follow-up. Median DOR was 8.4 months and 18.6 months respectively in these 2 cohorts. Here is a typical case of both MET amplification and overexpression showing primary resistance after 4-week monotherapy. However, he also achieved dramatic partial response just after 4 weeks of the crossover treatment with combination therapy, savolitinib and osimertinib. PFS data are not mature. However, median PFS was 9.3 months and 19.6 months, respectively, in these two cohorts. Talking about overall safety summary, equal or greater than grade 3 TIAEs occur in 8.7 and 57.1% patients of osimertinib monotherapy cohort and osimertinib plus savolitinib cohort, respectively. TIAE summary: treatment-related safety profiles of osimertinib monotherapy and osimertinib plus savolitinib were consistent with previous reports. Most TIAEs were grade 1 or 2. The most common TIAEs were diarrhea, rash, and pruritus in the osimertinib cohort, and rash, thrombocytopenia, peripheral edema, and ALT increase in the osimertinib plus savolitinib cohort. No fatal AE was observed in both cohorts. Conclusions: FLOWER study, CTON 2008, is the first phase 2 prospective randomized study to report efficacy and safety data of osimertinib with or without savolitinib as first-line treatment in de novo aberrant EGFR mutant advanced non-small cell lung cancer patients. Combination therapy with osimertinib and savolitinib demonstrated clinically meaningful improvement in primary endpoint OR. Immature PFS data also show a positive trend in favor of combination therapy of osimertinib and savolitinib. Safety profiles of osimertinib monotherapy and osimertinib plus savolitinib are as expected, tolerated, and manageable. This combination regimen has potential to provide a novel first-line treatment option for such patients. We thank all the patients, family members, and caregivers. Also thank all the investigators, nurses, and study staff members who participated and supported this trial. Thank you very much indeed for your attention.
Thank you very much, Dr. Yan. So, any question from the floor? No question? So, if no question, I just like to move on. Next, our presenter is Dr. Xiangjiao Meng. Yeah, Dr. Meng is from the Shandong Cancer Hospital Institute, Shandong First Medical University. Dr. Meng is Director of Fourth Ward of Chest Radiotherapy at the hospital. Dr. Meng, please.
Thank you, Professor Zhou. It's my pleasure to present the interim analysis of the phase 3 POLESTAR study. The leading PI is Dr. Jiming Yu. Omilatinib after chemoradiotherapy in unresectable stage 3 non-small cell lung cancer with EGFR mutation. Consolidation therapy with durvalumab is established as a standard of care for patients who do not experience disease progression following CRT. However, the specific benefit of consolidation immunotherapy for patients with EGFR mutated non-small cell lung cancer remains uncertain. Omilatinib, a third EGFR TKI, has demonstrated established efficacy in both first-line and second-line treatment of EGFR mutated non-small cell lung cancer. This phase 3 POLESTAR study aims to assess the efficacy and the safety of omilatinib after chemoradiotherapy in stage 3 unresectable EGFR mutated non-small cell lung cancer. Study design and treatment. Key inclusion criteria: 18 years or older, ECOG score 0 or 1, EGFR exon 19 deletion or L858R, locally advanced unresectable stage 3 non-small cell lung cancer, no progression during or following CRT. The patients were randomized at a 2-to-1 ratio to omilatinib or placebo until disease progression, toxicity, or other discontinuation criteria crossover is allowed for patients in the placebo arm when progression assessed. Primary endpoint, PFS assessed by BRCR. Sensitivity analysis, PFS assessed by investigator. Secondary endpoint including OS, ORR, DOR, safety, et al. Between April 28, 2021 and February 5, 2024, 147 patients across 43 sites in China were enrolled and randomized. The modified intention to treat set for efficacy assessment included 92 patients in omalatinib arm and 50 patients in placebo arm. The baseline characteristics including age, gender, smoking status, histology, ECOG score, staging, CRT type, concurrent or sequential, EGFR mutation were all well balanced in the two arms. The response to prior CRT was 60% in omalatinib group and 46% in the placebo group. Median follow-up of PFS was 16.36 months for omalatinib and 13.93 months for placebo. The median PFS was 30.4 months for omalatinib, while it was only 3.8 months for placebo. P-value was less than 0.0001. PFS-HR by BICR analysis with Cox methods was 0.200, indicating an 80% reduction for the risk of disease progression or death. The 12 months PFS rate was 69% for omalatinib and 21% for placebo. Investigator-assessed PFS results were consistent with the findings of BICR. PFS-HR analyzed with Cox methods was only 0.150, indicating an 85% reduction for the disease progression or death. PFS benefit favoring omalatinib was consistent across all predefined subgroups, ranging from 0.14 to 0.40. The ORR and the DCR were higher with omalatinib than with placebo. Median OS was not reached in either group. Lower incidences of CNS lesions and distant metastasis were observed in omalatinib arm than in the placebo arm. The most common AE for omalatinib was blood creatinine phosphokinase increased. Majority of the AEs were less than grade 3. Radiation pneumonitis was reported in 45% for omalatinib and 30% for placebo, with none being grade 3 or higher. Interstitial lung disease were not reported for omalatinib, but reported in one patient for placebo. AE leading to treatment discontinuation was only reported in 2.1% with omalatinib and 1.9% with placebo. Conclusions. In POLESTAR study, omalatinib exhibited a significant improvement in PFS compared to the placebo. Median PFS was 30.4 months for omalatinib, 3.8 months for placebo, HR 0.200, p-value was less than 0.0001. PFS benefit for omalatinib was consistent across all predefined subgroups. Furthermore, the overall safety profile of omalatinib after chemoradiotherapy was well tolerated and manageable with no new safety signal. These findings demonstrate omalatinib as a novel treatment option for patients with unresectable stage 3 EGFR mutated non-small cell lung cancer after the CRT. Thanks for your attention.
Thank you, Dr. Mong. Any question, please? So did you check the core mutation such as TP53 and etc. in your study? TP53 mutation? No checked?
53, yeah. We just enrolled the patients with 19 deletion and exon 21
Okay.
L858.
The EGFR sensitive mutation, right?
Yeah.
Okay, good. Thank you, Dr. Mao. Our next presenter will be the Professor David Blanchard from Institute in Gustave Roussy and Dr. Xiulin Lu from Emily Anderson. Dr. David Blanchard is thoracic oncologist, the head of thoracic pathology committee at the hospital. He has involved so many clinical studies, leading so many trials. Quite famous. Dr. Blanchard, please.
Thank you so much for the invitation. So it's a pleasure to have this presentation and on the further trial, which is a global randomized trial of fumonitinib at 2 dose level in TKI-naive advanced non-small cell lung cancer with specific mutation, the EGFR PAC mutation. So you may know that the EGFR P-loop and the C-helix compression, also called the PAC mutation, occurred for around 12% of EGFR mutation, mainly located in exon 80 to 20. And the PAC mutations are quite similar to the exon 20 insertion in narrowing the drug binding pocket to affect TKI activity. This PAC mutation can exist as a single PAC mutation, but also compound PAC mutation, either with an association of PAC or other EGFR mutation. And currently, in the real-world data, there is no standard of care in this population, first line, with a PAC mutation. So fumonitinib is an oral, highly CNS-penetrant EGFR inhibitor with a broad activity and selectivity across EGFR mutations. It has been currently approved in China, first line for the classical mutation, exon 21 and deletion 19, but also in the refractory patient with a mutation of resistant, like the T790M mutation. In the FAVOR trial, which focus on EGFR exon 20 insertion, patients receive fumonitinib 240 milligrams in first line of treatment, and it has been shown a promising response rate with 78.6%. And currently, fumonitinib have received FDA breakthrough designation therapy in first line for this population with an EGFR exon 20 insertion. Fumonitinib and its major metabolic, the AST5902, are both highly active against the EGFR PAC mutation, as you can see the figure on the right with different cell lines, highly potent activity against PAC mutation with both fumonitinib and the metabolite, while it spares the EGFR wild type at the dose which is currently used in clinical practice with fumonitinib. On the left, you can see also the unique structure of fumonitinib, which accounts a specific trifluoroetoxy group that will increase the contact with the binding pocket protein and hydrophobic residue of EGFR, including the PAC mutant that might explain the higher efficiency in this type of PAC mutation EGFR. So the further trial is a randomized trial. It's a global trial, phase 1b, with different cohorts, included one cohort, the cohort 4, which included only patients with an EGFR PAC mutation. Patients should not have received any previously EGFR TKI, and patients with brain mets, asymptomatic, untreated brain mets, were eligible to be included in the trial. Patients were randomized 1-to-1, either to receive the low dose level of fumonitinib 160 milligrams or higher dose level of 240 milligrams. Patients were assessed every 6 weeks. The primary endpoint was overall response rate according to the independent central review. Secondary endpoint, duration of response, CNS response, PFS, overall survival. Patients were recruited among 40 different sites, among 10 different countries. In terms of disease characteristic, it was similar between both dose level cohorts, and most of the patients were included in first line of treatment. Median age of this population with a PAC mutation EGFR in first line was 67, slightly higher proportion of female. You had 30% of patients with the brain mets at baseline, and nearly 2/3 of patients were never smoker or as young patients in this clinical trial. And this is probably one of the most important in terms of response. First-line PAC non-small cell lung cancer, according to the independent central review, the best overall response rate, which include the confirmed and the unconfirmed response, at the lower dose level was 47%, and it reached 81% at the highest dose level. In terms of confirmed overall response rate by independent central review, the lower dose level, 34% response rate, it reached 63.6% at the highest dose level of 240 milligrams, and disease control rate, which include partial, complete, and stable disease, was 100% at the highest dose level. And the assessment done by the investigator was quite close to have been observed in the independent review committee. Waterfall plot that show you the tumor shrinkage according to the dose level, the left 160 milligrams, on the right 240 milligrams. A confirmed partial response was observed in a wide range of EGFR PAC mutations, including the most frequent EGFR PAC, which are the G719 or the S768, as well the less frequent PAC mutation. And the response were also observed in the single and compound EGFR PAC mutation, and you can see on the waterfall plot, patients with a compound mutation are shown with a small star, and nearly 50% of patients in this trial had a compound PAC EGFR mutation, so highly potent, highly efficiency of fumaratinib in this compound PAC mutation. In terms of duration of response, currently the median follow-up is short, only 4.2 months, so the median duration of response currently has not been reached, as 20 of 22 patients of responder are still on treatment, PFS, OS immature, and as you can see on the figure on the left, it's a triangle, yellow triangle. Generally, the response have been seen at the first tumor assessment in the vast majority of patients in this trial. Brain mets, just to remind, so patients asymptomatic, untreated with brain mets were eligible in the trial. In terms of confirmed CNS overall response rate, at the lower dose level, 55.6%, higher dose level, 42.9%, and in first line of treatment, the CNS overall response rate, 46.2%, so highly efficient drug on the brain mets. You have an example on the right, a patient in first line of treatment, brain mets, untreated brain met that received fumaratinib 160 milligrams once daily, and you can see at week 24 a nice CNS tumor shrinkage of 82% in this patient in first line of treatment. In terms of safety profile, it was concordant what had been observed in the PAC cohort patient in comparison to the all patients included in the further trial, which included 158 patients. Just to mention, in terms of treatment-related adverse event of grade 3 or higher, it was 12.9% at the lower dose level, slightly higher at the dose of 240 milligrams, 20.7%. Dose reduction was 24% at 240 milligrams, and no dose discontinuation have been observed in two dose levels. And the treatment-related adverse event of clinical of interest were mainly grade 1 or grade 2. In terms of digestive toxicity, diarrhea, liver enzyme elevation, skin toxicity, the rash, dry skin, nail toxicity, and around 20% of stomatitis have been reported. Again, mainly grade 1 or grade 2 toxicity. No grade 4 or 5 have been reported, and no pneumonitis or ILD in this cohort have been so far reported, so clearly a safe drug. So that means currently it's highly promising anti-tumor activity in this specific highly selected patient with a PAC mutation. You have seen at 240 milligrams the confirmed overall response rate, 63.6%. Also encouraging CNS activity, acceptable and manageable safety profile. So clearly this drug need to go further development, particularly for this population, EGFR, PAC mutant, non-small cell lung cancer. And with this, I want to thank for sure the family, the patient. I want to thank all the investigators and the pharma that sponsored the trial. Thank you for your attention.
Dr. Blanchard, any question? So that's very good study. The name of study is further. What's the next step? I'd like to hear about further.
Correct, this is the question. So we're discussing between the pharma to know what to do. So that means probably the best things will be to have a randomized trial to confirm the benefit, and we might imagine to have a comparison with chemotherapy, particularly to get access to the market, and if we want to get access to the market in Europe, otherwise they might get access and approval, access to the market perhaps for the North American, for the FDA, for the EMA, it's much more challenging, and probably for ASEAN, but currently it's discussing.
Okay. Dr. Li, any comment?
Yeah, so I agree with David Blanchard. We're planning going forward will be randomized global study. Interestingly, because PAC mutation is such a understood disease space and is such under-investigated mutation, so the practice currently is very heterogeneous. In Europe, in China, and other parts of Asia, and in US, I think physicians pick different standard of care choice. So the randomization to which control arm is still an active discussion. Nonetheless, this is the first EGFR TKI being evaluated in the PAC mutation. PAC mutation is more common even than EGFR exon 20 mutation. So we feel very encouraged by this data. We felt that we might be able to provide something more homogeneous and more potent with CS efficacy to this patient population.
I have a last question. You know, formotinib is not available outside China. It's only available in China, right? When you see a patient with PAC mutation, do you like to treat them with osimertinib at double or triple dose?
I would say when I see the result, probably I would be confident to treat this patient at 240 milligrams. When you see the magnitude of benefit and particularly the response rate, you reach 63% in terms of overall response rate. So we need to have further data, particularly in terms of duration of response, PFS, and so on. It's currently immature, but I'm quite confident. And finally, the ratio in terms of toxicity is quite fine because it's mainly grade 1, grade 2 toxicity, and we know how to manage this type of toxicity. And I think it's close what we observe with osimertinib with an excellent tolerability.
Okay. Thank you very much, Doctor.
Thank you so much.
Dr. Blanchard. Next presenter will be Dr. Xiaoning Li. She is the tenure-track assistant professor in Department Thoracic and Head Medical Oncology at MD Anderson Cancer Center. Dr. Li has accomplished so many clinical studies and trials. Dr. Li, please.
Yeah, thank you, President, for your nice introduction. On behalf of my co-investigators, I present the safety and efficacy of BEAR-88 in patients with HER2 mutation non-small cell lung cancer patients, expansion cohort D from phase 1/2 trial, SOHO-1. HER2 kinase domain mutation occurs in about 3% of all non-small cell lung cancer. Those mutations are oncogenic and can be inhibited by small molecule inhibitors. The most common insertion mutation for HER2 is YVMA at 772, occurring about 75% of the mutation. Currently, chemotherapy and ADC are the approved treatment for this HER2 patient population. Therefore, there is a great unmet need to have an oral medication that's highly efficacious and have CNS activity to help this patient population. BEAR-88 is oral reversible small molecule inhibitor targeting HER2. Preclinical studies demonstrated excellent efficacy against HER2 mutations, including YVMA. SOHO-1, the first part of the study, already demonstrated preliminary efficacy and the safety in HER2 patients. Therefore, BEAR-88 currently has already received the US FDA as well as Chinese NMPA breakthrough designation. SOHO-1 is a global multi-center, multi-cohort study having dose escalation and dose expansion parts. At the dose escalation part, we identified BAR88 20 milligrams twice a day to be the recommended expansion dose. In the expansion parts, we have different cohorts evaluating different genotype and different patient characteristics. The primary endpoint for the entire study is safety and tolerability. The secondary endpoint is response rate by investigator assessment. I presented cohort D at today's presidential symposium. This is a cohort enrolling HER2 mutation patients with total exon insertion. Patients have received prior systemic therapy, but they have never received a prior anti-HER2 treatment. A total of 44 patients were enrolled. Majority of the patients are female, never smoker. Majority of the cancers are adenocarcinoma. HER2 YVMA represents 70% of the cohort, and the brain metastasis at the baseline is around 20%. Overall, this is a very consistent data to our knowledge of HER2 patient population in lung cancer. The efficacy in cohort D, the response by investigator by RECIST 1.1 was 72.1, including one complete response. Disease control rate is 83.7%. As indicated on the waterfall plots, majority of the patients have tumor shrinkage and the benefit regardless of genotype or brain metastasis. The response were achieved early, mostly at the first assessment. At the median follow-up of 10.9 months, 16 patients are still on treatment, including 14 patients at treatment duration greater than 1 year. Median duration of response is 8.7 months, and the median progression-free survival is 7.5 months. HER2 YVMA is an important subgroup because it's the most frequent insertion mutation. In our cohort, we had 30 patients. In this population, response rate was 90%. Disease control rate was 97%, median duration of response 9.7, median PFS 9.9 months. In addition, patients with brain metastasis and patients who received prior immunotherapy all derived a similar magnitude of benefit compared to the total cohort. In this cohort, BAR88 demonstrated a manageable toxicity profile. Diarrhea remained to be the most common treatment-related AE, mostly in grade 1 to 2. There is no safety signal. 7%, 3 patients had treatment-related AE leading to dose discontinuation of BAR88. So in summary, BAR88 treatment lead to rapid and durable response in patients with pretreated HER2 mutation. The safety profile of BAR88 was manageable. This data supports ongoing investigation of BAR88 in patients with advanced non-small cell lung cancer with HER2 mutations. With this, we thank the patients and their family for their participation. We thank our global investigators, and we thank our sponsor for their commitment. Thank you.
Yeah, thank you very much, Dr. Lee. Any question? So for HER2 mutation, now we have TDXD, which is available in the market. So now you report the data of the HER2 TKI. What's the future of the two compound? You like your compound to first line or second line?
Yeah, thank you, Dr. Zhou, for this question. I think I want to highlight 2 points. One is small molecule inhibitor and ADC, because the mechanism of actions are so different. Yeah. So one is the antibody drug conjugates is antibody-based approach, but small molecule targeting the ATP pending pocket. So I think our patient will need both options and as fast as possible. And then those medications, from the mechanistic standpoint, they probably can be sequentially used because the resistance to those 2 different class of drugs can be quite different. Yeah. So I think number one, we need TKI regardless, because currently we don't have any TKI option. The second point I want to make is that I do hope that the TKI can move to the front line, in that is oral medication is more convenient for the patients if we can have the toxicity being managed. As the example of EGFR patient population, I think patients enjoy really high quality of life with oral medication up front for long duration. We hope over time we can start to get there.
So in your study population, any patient previously treated with TDXD?
No, so in our cohort D, we only had patients had prior chemo or immunotherapy. And in this cohort, none of the patient has received any anti-HER2 targets, either ADC or small molecule. Okay. But in the SOHO study, we do have a cohort dedicated for patients who had prior TDXD, no prior TKI. Hopefully that cohort will be read out soon.
Okay. So how about combination TDXD and Bear 292708 compound together in front line?
Yeah, I think that's actually a great question, because just like I mentioned earlier, the mechanism of action, the ADC and the small molecule, they're very different. So one can imagine that the ADC drug bind to the membrane, and then the TKI go into the ATP binding pocket, and then have a truly synergistic, on-target, deep inhibition, so that the tumor shrinkage and duration can be fantastic. We need to be mindful of the on-target toxicity as well, but I think that type of combination definitely need to be evaluated both preclinically and clinically.
Okay, thank you very much, Dr. Li. Next presenter will be Dr. Galina Lotter. Dr. Lotter is pulmonologist, specialized in the thoracic oncology in the Netherlands Cancer Institute in Amsterdam, Netherlands. She works in the Department of Thoracic Oncology and Department of Clinical Pharmacology, focus on early clinical drug development and phase 1 study. Dr. Lotter, please.
Thank you for the introduction. So I'd like to present on behalf of the team the results of the phase 1b analysis of Beamion LUNG-1, zongertinib in patients with HER2 mutant non-small cell lung cancer. As Dr. Li already told us, HER2 mutations are rare in non-small cell lung cancer and are associated with a poorer prognosis and higher incidence of brain metastases. Zongertinib is a third-generation orally administered HER2 TKI that selectively and covalently binds to the tyrosine kinase domain of both wild-type and mutated HER2 receptors, but spares the wild-type EGFR and thereby avoids associated EGFR toxicity. The Beamion LUNG-1 trial is actually a classic trial design with a phase 1a dose escalation part and a phase 1b dose expansion part. The phase 1a was for all solid tumors with HER2 aberrations, and phase 1b was solely for non-small cell lung cancer in different cohorts, as you can see here on the slide. From phase 1a, the 120 milligram and 240 milligram doses were selected as the recommended doses for expansion. And in phase 1b, cohort 1, which was the cohort that we presented today, pre-treated non-small cell lung cancer patients were included, having a HER2 mutation within the tyrosine kinase domain. They were randomized to 120 milligram or 240 milligram for the FDA Project Optimus. And after interim futility analysis, the 120 milligram dose was selected for further evaluation. These are the baseline characteristics of cohort 1. 132 patients were included at the data cutoff in May 2024. And the baseline characteristics are quite well balanced. I'd like to highlight the two things that have green boxes around it. Actually, well, the first box is around the first line, but I would like to emphasize that most patients have more than one line prior treatments. So a quarter of them actually have 3 or more prior treatments before starting on zongertinib. And many patients have brain metastases. So patients with asymptomatic, untreated brain metastases were allowed to enter the study. These are the main safety findings. In general, the majority of treatment-related adverse events were mild and manageable. And as you can see, diarrhea and rash were the most common adverse events, as you would expect from a HER2 TKI. But importantly, only 2 patients had grade 3 diarrhea at any point during the treatment. And most patients had grade 1 diarrhea. And to emphasize, we always talk about, is it manageable or not? This diarrhea is actually quite good manageable. You give them loperamide, maybe stop the treatment for a few days, and the diarrhea will disappear. So it's generally well, they do well. There were no fatal treatment-related adverse events, and the adverse events that led to dose reduction occurred in only 11% of patients. 4 patients had to stop the treatment because of adverse events, and these adverse events were ALT and AST increase. Then the tumor response. The primary endpoint, which was a confirmed response by BICR, was met for all treated patients at the 120 milligram dose. The overall response rate was 66.7%, but several ongoing patients could still achieve response, so maybe this number will be higher in the future. As you see in the waterfall plots on the right bottom of the slide, tumor shrinkage of any magnitude was observed in 94% of the patients. The duration of response and the PFS data are currently immature because 2/3 of patients were still on treatment at the data cutoff. We actually expect this data to become available by the end of this year. And in the table below, the patients are shown that were randomized to the different doses. And as you can see, in those patients, the response rates are even a bit higher, and also with very high disease control rates. This is a patient case I'd like to show. It's a 67-year-old female having received second-line zongertinib after progression on chemoimmunotherapy. She progressed, she did not progress. She responded quickly. With resolve, her complaints disappeared, and the response was already seen after 6 weeks on the first CT, but clinically only after a few days already. And the best response here is shown on the slide after 5 months. This patient is still ongoing on the trial, which was 13 months at the moment we send in the slides. This is a slide of the intracranial response. Although the numbers are low, we do see encouraging preliminary intracranial activity with zongertinib. In the 120 milligram dose, it's 33% with high disease control rates. So again, these were asymptomatic, untreated brain metastases that were allowed to participate in the study. And on the MRI scan on the right of the slide, you can see that one of the metastases that is most easily seen, also in the back of the room, disappeared after only 6 weeks. The duration of intracranial response for this patient was 6 months. To conclude, zongertinib demonstrated clinically meaningful activity in patients with pretreated non-small cell lung cancer with a HER2 mutation within the tyrosine kinase domain, including in those with brain metastases. Zongertinib was very well tolerated. No deaths were attributed to treatment, so no grade 5 toxicity, and the low incidence of dose reductions and treatment discontinuations. 2/3 of patients are still on treatment at the data cutoff, and the FDA has granted breakthrough therapy designation. And now the Beamion LUNG-2 trial is enrolling, which is a phase 3 randomized trial of zongertinib compared to standard of care as a first-line treatment for this patient category. Many thanks to all the patients, of course, for participating, the families, and the clinical study team all around the world. And thank you for listening.
Yes, thank you very much. Any question? So there are so many variants of HER2 mutation. How about the compound efficacy to different variants?
I'm sorry, can you repeat the question? A different
variant, different HER2
Oh,
mutation.
different variants.
Yeah.
I can maybe show this one. So as you can, it's not on the slide anymore, I'm so sorry. But the YVMA mutation was also in our population, the most prevalent one, which you would expect is actually the same as Dr. Lee showed. Yeah. Our numbers are a bit lower than what she discussed, but these are the most common ones.
Okay, good. Yeah, thank you very much. So, thank you very much.
Okay, thank you.
Finally, as our customer here at Yes Lung, we invite one of our patients advocates to provide unique patient perspective on the research presented today. ISLUG is pioneer among the research association including patients in our programs, events, and press briefing. Please welcome patient advocate Angus Brett. Angus is diagnosed with male breast cancer in May 2018 in the workshop for treatment. Primary stage 3C lung cancer was discovered. Initial aggressive radiation therapy was followed by nivolumab immunotherapy until after 9 months progression observed. In the meantime, Angus had bilateral mastectomy for his stage 1 breast cancer. His lung cancer was EGFR positive. On the targeted therapy with afatinib, he remains stable for up to this today. Angus was involved in patient advocacy as mentor in Cancer Connection.ca. He helped with several patient-driven surveys with Lung Cancer Canada around COVID-19, as well as participating in the Wrong Question campaign. Mr. Angus Pratt, please.
Thank you, Dr. Chow. What a privilege to be able to respond to all of this magnificent research. I stand here as a breast cancer patient, and as you can imagine, I heard a lot of HER2 discussion in that whole field. And I think one of the things that really strikes me is that as we approach sort of the solid tumor piece, we're starting to understand that some of these genetic drivers cross over between the solid tumors, and a lot of the treatments and a lot of the advances and the vast amount of money that's spent on breast cancer is going to finally trickle over and have some reflection, I think, in lung cancer as well. Though the fact that I'm HER2 positive with my breast cancer does not mean that I'm HER2 with my lung cancer either. So that's my first comment. Just amazing to see that kind of work happening here. When I started treatment 6 years ago, HER2 was not on the horizon at all. And so to see and hear some of the discussion is exciting. The second comment that I would make is as I listen to all of this, I'm still struck by one of the discussants made the comment that 43%, about 50% or 50% of people have no biomarker testing before they begin treatment. And all of this work is based on biomarker testing, and it is so critical that we advocate and make sure that patients have biomarker testing done before they begin treatment. It does take time. There are mechanisms that are shortening all of that, but it still needs, it's a question that needs to be addressed, and more oncologists need to be aware of the fact that biomarker testing is critical. Another overarching fact that really struck me about the variety of work here is for me a relatively new focus on CNS progression or CNS inclusion in looking at the results. I think that that's a relatively new development. Lung cancer has primarily been focused on lungs, but we know that the first place it goes, or often the first place it goes, is the brain. And so, and that is a critical organ I've discovered in order to operate well. My final comment would be just, I'm really excited as I sit here and think about the variety of treatments that have been presented here today. The options for lung cancer patients are growing, and we're seeing so many exciting new novel treatments. Yes, these are probably 3, 4, 5 years from a market and availability to patients, but those who are coming behind me will need these, and it's just an exciting moment to be involved and excited about the options that are becoming available. Thank you all for your hard work for us.
Yeah, thank you. Thank you very much, Angus, for your excellent sharing your perspective and a moving life story with us. So last, any question from our floor to our panelists? No. If no question, I like give my thanks to all the panelists, experts for your excellent presentation and discussion, and also for your excellent work. Thank you very much. I'd like to close this section, please.